So today my presentation will focus about our main results of our last article published in Eno1 and in IVES technical review entitled New Insight about Sensory Contribution of Grape Stems During Winemaking, Role of astilbin, a Sweet Polyphenol. So when you taste a wine, it involves several of your senses, namely sight, smell, taste, and touch. And the sensory quality of a wine will depend on its aromatic typicity, but also on its gustatory balance. And in dry wines, this balance is based on saltness, bitterness, and sweetness. Concerning sweetness, in dry wine, this taste is not due to the presence of the residual sugar. In fact, they are present below their taste detection threshold. So we cannot perceive them. So the sweetness is therefore due to the presence of other non-volatile compounds. In fact, the taste-active compounds come from different origins in wines, as in grape, fermentation, or even in oak wood. And our laboratories work for several years to identify the sweet compound in wines. So we identify some sweet compounds come from the grapes as EPDPAG and astilbin. We also highlight the contribution of protein HSP12 with its sweet peptide. And more recently, we identify a new sweet molecules. The N6-succinyladenosine comes from the yeast autolysis. And there is also some sweet molecules extracted from oak wood, like quercetriterpenosid and dihydroconiferyl alcohol. But today, my presentation will focus on astilbin, which is a dihydroflavonoid come from grape. In fact, we do some sensory analysis about this compound, and we do gustatory analysis in hydroalcoholic solution, but also in wine. And we found that these molecules has an intense sweet taste. We can also quantitate these molecules in wines. So here I represent you some results about the quantitation of astilbin in 123 commercial red wines. And we calculate an average of 9 milligrams per liter of astilbin in this wine. And its concentration can vary between 0.5 milligrams per liter to 56 milligrams per liter in certain wine. We measure also its detection threshold at 5.7 milligrams per liter. So that means here for several wines with content higher than this value, in this one, astilbin can participate directly to its sweet taste. With this result, we can also study the influence of the grape variety. And as you can see here, we found more astilbin in wine made from Gamay and Pinot Noir than wine made from a blend of Cabernet Sauvignon and Merlot. And which is interesting to note is for Gamay and Pinot Noir, sometimes these wines are vinified with the presence of stem. And as you know, during the winemaking process, there are several steps. So after harvesting the grape, the bunch are the stem, and crushing, and the juice will send to veg. There is a stage of waiting before the running off. But sometimes the bunch are not the stems, and we call that the whole bunch vinification. And wine growers can also destem the bunch and can after add some stems during the vinification protocol. And the works of Blandine Crétin during her PhD, she works on the localization of the sweet compounds in various constituents of the Merlot bunches. And she showed that 97% of the total quantity extract of astilbin came from the stem. So we therefore looked if this practice, so the whole bunch vinification, had maybe an impact on the astilbin content. There is some study in the literatures who worked on this practice. They mention some aromatic characteristic with a vegetal aroma, but also some of them study the gustative characteristic with bitterness and astringency. However, these studies, most of the studies have compared wines made from destem grapes and wines made from 100% of whole bunches. Concerning our experiments, we do this experiment in real winemaking conditions in different estates during different vintage in 2019, 2020, and 2021. We do this experiment in different French regions, in Bordeaux, and in regions where this practice is more common, in Beaujolais and in Bourgogne. So we study different grape varieties, Merlot, Cabernet Sauvignon, Gamay, and Pinot Noir. And we use different percentage of whole bunch according to the estates, namely 15, 30, and 50% of whole bunches. Sometimes one grower decided to add the whole bunches directly, and sometimes they calculate the stem to add during the weighting protocol. So we have our modality with stems, and for each vat with stem, there were another vat without stem, and we call it destemmed. So we have two modalities, and we do a total of 13 experiments. In the literature, some studies focused on the acidity and on the alcoholic content. And concerning our studies, adding stem in the proportion used in our experiments had no effect on the pH or on the alcoholic content. Now, concerning astilbin, as you can see here, we found more astilbin with modality with stem, with here an average of 24 mg per liter, compared to the modality destemmed with an average of 20 mg per liter. So here, adding stems in the proportion used in this experiment increased the concentration of astilbin. Now we can also follow the extraction of astilbin during the weighting process. So I show you different examples here in three grape varieties, in Gamay, Pinot Noir, and in Merlot. So as you can see, the extraction of astilbin occurs during the first few days of the alcoholic fermentation, and vats with added stem contain more astilbin. But which is interesting to note is the difference between the modalities. So here for the experiment in Gamay, the difference between destemmed and 13% of whole bunches is about 15%. Concerning the Pinot Noir, the difference between the two modalities, destemmed and 50% of whole bunches, is about 21%. And finally, for Merlot, the difference between destemmed and 30% of whole bunch reached 41%. In fact, we calculate this mean difference. So we calculate the mean concentration of astilbin in each modalities according to the grape variety. And so we found more astilbin in Gamay and Pinot Noir that I presented before. But in fact, the difference between the two modalities is higher in one made from Merlot than in one from Gamay and Pinot Noir. And if we compare with the detection threshold, the modality with stem become higher than this detection threshold. So we try to understand where this difference comes from. So we quantitate astilbin in the stem of this different grape variety. And we found more astilbin in Merlot here than in Gamay with a significant difference. So finally, for Gamay and Pinot Noir, astilbin can be provided by other constituents like skin, seed, and pulp. That's why we study these different constituents, and so we continue the study with 30 bunches. So after the separation of these different constituents, we have freeze-dried these constituents and macerate them to quantify them by LC-HRMS. And here I represent you the mean concentration of astilbin in this constituent. So we found very few microgram per gram of astilbin in pulp and seeds. And we found on average 100 microgram per gram of astilbin in skins and more than 400 microgram per gram astilbin in stems. So finally, here we demonstrate that stem was a constituent containing the most of astilbin. But finally, in a bunch, there is different proportion of this constituent. So after separation, we weighted them individually. And on average, we calculate the average proportion of this constituent, and we found 76% of pulp, 14% of skins, 6% of seeds, and only 4% of stem. So now I calculate the mean constituent of astilbin for each constituent within the cluster. So we found, of course, very few microgram per gram of astilbin in pulp and seeds. And now concerning the skins and stem, within the bunch, astilbin was located at 6 microgram per gram on average in skins and 5 microgram per gram on average in stems. I represent you this result also in proportion. So finally, we can see that astilbin is divided equally between skins and stem. So now, among our 30 bunches, there was 10 bunches come from Gamay, 10 from Pinot Noir, and 10 from Merlot. So we can compare this grape variety also. So we found that Pinot Noir bunches contain on average twice as much astilbin than Merlot bunches. And which is interesting to see is the proportion of astilbin located on this constituent according to this grape variety. So concerning the skin, the proportion of astilbin is higher in the skin of Pinot Noir grapes than those of Merlot grapes. And concerning the stems, the proportion of astilbin is significantly higher in the stem of Merlot grapes than those of Gamay and Pinot Noir. So finally, we can say that for Pinot Noir and Gamay, astilbin is located of a half in stem and another half in skins. And concerning Merlot, we found 2/3 of astilbin in stem and 1/3 in skins. So these results help us to more understand the difference in ratio between the two modalities with and without stem during the winemaking process. I will show you other parameters that can influence the astilbin concentration in wine. So we study the same Bourgogne wine depending on the vintage. We quantitate astilbin in the stem and in the corresponding wine. And we found a global lower concentration of astilbin in 2021. So we look at the precipitation between April and harvest. And here you can see that the lower stress is a priori lower in 2021. We also do other experiments with the measurement of isotopic ratio of carbon-13 in several Bordeaux wines. And finally, we showed that there is an interconnection between the water stress and the astilbin concentration. We also followed the evolution of astilbin over time. And we quantitate these molecules in several vintages of a Pinot Noir wine from the same estate from 1918 to 2020. And you can see in this graph that astilbin tends to be higher in young wine than in old wine. In fact, we demonstrate that these molecules isomerize over time in different forms in neo-isoastilbin, neoastilbin, and isoastilbin. And we quantitate also these other isomers. And we found that this other isomer, especially neoastilbin, is higher in old wines than in young wines. And the trans isomers are more abundant in very, very old wine. And so we studied also their taste, and we found that all these isomers are sweet with different intensity. And we found that neoastilbin and neo-isoastilbin are sweeter than astilbin. So we also studied the gustatory effect of astilbin and the mixture of isomers. So we compared a solution of astilbin alone with a solution of mixture of this isomer in different proportion. So with the triangular test, we showed that there is a significant difference between these two solutions. And we asked the panel to evaluate the sweetness, and we found that the solution of astilbin and the solution of mixture of isomer are sweeter than the control solution, so without addition. And here, even if the difference is not significant between astilbin and mixture isomer, but mixture isomer seems to be sweeter than astilbin alone. So to conclude for this presentation, we work on the effect of whole branch vinification on the astilbin content. So the addition of stem had no effect on pH and the alcoholic content, but it increased the level of astilbin. And there is some variation according to the grape variety. In fact, the ratio between the two winemaking methods is higher in Merlot than in Pinot Noir and in Gamay. We also studied the localization of astilbin is different part of the bunch. And we showed that for Pinot Noir and Gamay, astilbin is divided equally between stem and skins. And concerning Merlot, we found more astilbin in stems than in skins. And it contributes to explain the difference in ratio between Pinot Noir and Gamay with Merlot. Concerning the study of the same Bourgogne wine depending on the vintage, we shown that there is an intern connection between water stress and astilbin concentration. Finally, concerning the evolution of astilbin over time, we demonstrate that the molecule isomerize over time into isoastilbin, neo-isoastilbin, and neo-astilbin, and the isomer mixture appears to be sweeter than astilbin alone. Finally, these results bring new tools to better understand the practice of whole branch vinification from a chemical perspective. Of course, the practice of whole branch vinification can also bring some undesirable compound like vegetal aroma or bitter astringent compound. So the use of this method must be carefully controlled, and the knowledge about this practice have to be further developed. Thank you very much for your attention.